So, medical informatics. Well, one of the questions I always get asked is, why are you doing medical informatics in the HBP? Because that's a sort of neuroscience thing. So you have to think about brain diseases as clinical neuroscience, and you have to think about brain diseases as everything from depression through to brain tumors. So that's the sort of, that's what we're going to be thinking about. And what I'm going to do today is to give you a bit of background as to why, what are the motivations for medical informatics in the HBP, then what's the rationale, what's the initial evidence that this might be a useful thing to do, because the only difference between basic science and medical science is that there's always that idea about being useful to patients in the medical side. Though, you know, even basic neuroscientists think about that sometimes. And then finally, I'm going to try and present to you where we've got with the medical informatics in terms of the infrastructure, which is the platform, which we've already deposited the prototype of, and which we have to deposit the internal release of at the end of March, which we're struggling to do, but we think we're going to do it. And I think, hopefully, that will be reasonably radical. I'm not the best person to talk about that, because that's being done by Anastasia Ailamaki and her team at EPFL. And the details of that in technical terms are more things you're going to have to look up on the site or write to Anastasia or whatever. And then finally, I'll show you some preliminary results. So that gives you a sort of idea. And you've had lots of talks about why we're focusing on the brain. Possibly many of you are motivated anyway because it's such a fantastic thing, this 1.3 kilograms that does everything a supercomputer does. It doesn't, of course. It does many things much worse than, but it does some things much better than, and it's certainly more energy efficient. So when we started this off with Henry Markram and Karl-Heinz Meyer, there were three bits that we were interested in. And this bit you're probably most interested in, and it's the sort of thing David and Karl-Heinz and many others have been playing with. And it really functions in two ways. One way is to support what we wanted to do, and secondly, to be inspired, possibly, by the architecture of the brain in terms of future architectures in computing. So that was the virtuous circle that we thought about. And the understanding brain diseases were justified by a number of things. One, that it's a social and economic challenge, much bigger than the HIV epidemic, probably as big as the Spanish flu epidemic, which, as you know, probably killed about 5% of the world's population in a year, and possibly as devastating as the old plague epidemics were, which, you know, at one stage killed 30% of the European population in a couple of years. So these are not trivial things, and it affects some sort of brain disease, affects 1 in 3 people in their life. So you know, there are quite a number of you here who are going to have brain problems. Actually, the funny thing is, what is the most expensive brain disease, do you think? Has anyone got an idea? If no one shouts anything, I will point at somebody. You. Yes. Alzheimer. No. Madam. Come on.
Depression?
No. God, Madam's changed her sex because it sounds very, very odd.
Glasses?
Not glasses, no. That's vision. That's a good idea, but it's something close to that. It's migraine. Migraine because it debilitates people for a short time, but recurrently. Migraine because when you have a bad one, you don't go to work, so that costs. Migraine because if you don't go to work, you may need support, you may need the doctor, you may need drugs, and so on. So all these issues come into the cost, but that's just by the by. It is the number one loss of economic productivity, so it's important to the politicians and to society in general. And for the big areas, which are psychiatry, on the one hand, and the degenerations of old age, we have nothing. And as I'll show you in a moment, not only do we have nothing, we don't even know how to diagnose them. So there are serious problems. So now, we have nothing. Neurology has existed since about 1864, when a gentleman called Broca, who was a professor at the Salpêtrière, turned up a patient, at Sainte-Anne in Paris. He turned up with a patient who couldn't speak. In fact, the only thing this guy could say is tongue, and this was something that had afflicted him suddenly. So it was likely a stroke. Blood vessel clotted off, bang, a lot of brain, can't speak. And the guy lived at Sainte-Anne for 6 years, up to 1964, and then died. Now, just to show you how quickly scientific information could get across in those days compared to the 2 years it takes to get a good paper into the literature these days, that night he did the autopsy, found a big hole on the left side of the head here, and presented it at the French Society of Neuropathology and Neurology the following morning, which completely changed neurology, because what it showed was that structure and function went together. And the whole of neurology since then has been based on that. It's been based on defining people who are ill in terms of two things. The first is what they say to you, their symptoms. I hurt here, I hurt there, tomorrow, yesterday, and so on. And the other thing it's defined upon is the doctor then forming a hypothesis about what's wrong with them from this verbal report, and then doing an examination of the body with the anatomy as the basis, as the template, and working out where there is something wrong in the anatomy and then putting the two together. And that's called a syndrome. So we have symptoms and syndromes. So as you see, they're difficult things to base treatment of people on, because when a patient comes into me with with her husband or with his wife and sits down, I already think to myself, they're either a very loving couple after 50 years, or perhaps there's something wrong that the other one needs to help me with. And then I say to one of them, well, hello, what are you doing here? What can I help you with? He says, I don't know. She told me to come. You've already got the diagnosis. Cognitive decline. Doesn't know there's anything wrong with him. Probably got Alzheimer's disease. Certainly got some sort of cognitive disorder. So the history all comes from the wife. So already it's a history by proxy. And, she may have been a nice, loving wife and give you a good, accurate history, or she couldn't have cared less. He was out in the pub all night. She was with the girls playing bingo. He says, well, he doesn't remember anything. We sent him out to shops. He didn't bring anything back. That's why he's just gone bonkers.
You are describing my marriage.
Yeah, I know. So this is quicksand, right? And then you come back to the examination. Well, the examination has improved very markedly over the last 40 years. Since 1972. Because in 1972, or up to 1972, we had to make inferences about the anatomy, certainly if there was anything wrong in the brain, mainly because you couldn't get into the brain of normal people, i.e. people who didn't have symptoms and syndromes while they were alive, to see what the normal brain looked like. You couldn't do operations on normal people. And so in 1972, the first CAT scanner arrived. And so we could suddenly see inside the skull without opening it up. And then you had the PET scanner in 78, and then you had the first clinical MRI scanners in 1989, so that now we can really look at the brains and the nervous systems of all normal people many times, because we don't use radioactivity, there's no tracers or anything, there's no x-rays. And we can look at the brains of abnormal people, so we could do what most scientists do, which is to compare and contrast normal with abnormal. So from about 1972, lots of new techniques came in which helped the analysis of the anatomy. So there we are. Now let me just drive this home for you. This is a very simple study, but one of those beautiful studies which just nails it down. This is a syndrome. It's called frontotemporal dementia of the behavioral variant. So it's a very specific diagnosis. It's based on pathology and lots and lots of studies with clinicians who've compared and contrasted, and so they've picked out this little set of symptoms and they think, right, that's it, we got it. We know how to diagnose this. And there are 32 of these patients here. It's a consensus criteria and everything. And the guy who did this is a guy called Kurtesh, a Canadian, a neuropsychologist. All he did was to follow these 32 for up to 10 years. And the amazing thing is that after 10 years, sorry, not after 10 years, after a mean of 3 years, you can see here 26 of them had a different syndrome. So this syndromic diagnosis here wasn't stable. It was stable in only 6 patients. And then when he went to pathology, there was no one single pathology that correlated with this syndromic diagnosis. Now, there may be reasons for that, but let's just stick with this. And then of these 26, after another 3 years or so, you can see there were still 12 more who went to yet another diagnosis, another syndromic diagnosis. Okay, and then when we take them all on to pathology, there's just no relationship one with the other. So depending on when the doctor saw the patient, that diagnosis would be given. Now imagine that you're a pharmacologist and you're looking for a drug to treat a precise disease with a precise pathogenic mechanism, which depends on a syndromic diagnosis. Well, you're stuffed, aren't you? And that's why, as somebody said in one of the lectures earlier on, the drug companies are pulling out of the dementias because it's quicksand. But no one really admits to this yet. They don't admit to it overtly because patients would be shit scared. The second reason that they don't admit it yet is that you can be demented and you could still be very powerful. So Mr. Regan in his second term was frankly demented, and you just need to listen to some of his speeches. It was quite clear that the government was being run by consensus, by committee, mostly probably by his wife and the Secretary of State, but he was still very presentable. He could talk on television, and even if he said words which didn't make much sense or which were new words, so invented words, people listened and people were impressed by him, and people voted for him. And people felt that he was one of the very few people who really, fought communism and won. So there are difficulties about doctors working with patients and relying simply on the syndromes and symptoms. Now, there are other problems. We were talking about pathology. If you look at the pathology at the end of a process that's gone on for 10 years, what you see is a shriveled little walnut weighing about 700 to 800 grams, starting at 1.3 kilograms. So it also depends on how you treat the thing. The brain, if you're going to look at it microscopically, you can't do the whole brain. Pathologists just look at the hippocampus, a little bit of brain here, which is known to be affected early in Alzheimer's disease. They haven't got time or money or storage space on computers and so on for looking at the whole brain microscopically. So there are problems. It's probably end stage. But the biggest problem of all really came out of functional imaging and was the very clear demonstration that this is an extremely redundant machine. So that, there are these people who go around saying, you only use 10% of your brain. Of course, complete rubbish, use the whole of your brain all the time. But the fact of the matter is you could lose, say, 70% of a particular set of cells, of which there are only 600,000 on each side out of the 10 to the 12 cells you probably have in your brain. When you lose 70% of the nigrostriatal cells, so 900,000 in total, you get the first symptoms of Parkinson's disease. You begin to notice your first tremble and your first bit of shuffle. So that means you can compensate the loss of 70%. And it looks as though in Alzheimer's disease, it's probably around 50, 60%. You can compensate for that. When you have a stroke, after, particularly if you're young and the stroke is in a deep place, you can come back to virtual normality over the next 3 or 4 months. So there is redundancy in the sense that there's the capacity to reconfigure the connections. Now, it may be that there are new synapses, or maybe there's new neurons, so that's very unlikely. But it's most likely that there are pathways which have lain unused, haven't been strengthened in the normal development, which now can be strengthened by, be it exercise or be it whatever drugs or whatever combinations of those that you can have. So, bad news for people who want to diagnose these diseases. Good news potentially for the patients, because if you can diagnose preclinical disease, in other words, while you're normal, don't forget we can't diagnose clinical disease yet. So here we are wanting to diagnose preclinical disease, but let's be adventurous. Then perhaps you could find treatments, if you knew the mechanisms, that would prevent the onset of the symptoms. Or even if they just slowed down some degenerative process, they might postpone them for, say, 5 years or 10 years. That might be quite enough to get you to the ripe age of 85 or 90, which is the average length span of your generation. So here we go. Now the final thing which I'm using Alzheimer's disease as a sort of paradigm here. Final thing I have to say about Alzheimer's disease is people really have tried to find out what causes it and so on. But the trouble is that we scientists are the most conservative of people. So what we tend to do is we tend to find something interesting in the literature or while we've been training or whatever, and then we find ourselves in a laboratory and we have certain sorts of equipment and facilities, and then we think, what can I do with these facilities that would help me from what I know? And people go for the easiest. So do all of you know about the amyloid hypothesis of Alzheimer's disease? Have you all heard about this? This horrible protein that clogs up the nerves and so on and so on. The beautiful thing about amyloid is the first thing you do in medical school when you go into a path lab is you get given a slice of brain and a tube of red liquid called Congo red. And you pour it over the slice, you wash it in distilled water. You don't even have to put it under the microscope, they make you do it, times 20. And you can see all the amyloid, birefringent crystals, or with the naked eye, red stuff. Oh, it's so easy to do, everyone can do it. So everyone's writing about amyloid. And now, after 20 years and roughly $10 billion of investment, everyone's saying, oh, perhaps this is an incidental finding or a tombstone of the pathology or whatever. So it really is important that we try and get some logic and some sense into these sorts of things. Now, neuroscience, basic neuroscience, should be helping in a big way. And as you can see, there are many, many, many papers being produced. This is a slide that Henry made and gave me, Henry Markram. And this is 1990, this is 2012. So in 2012, there were about 100,000 papers in neuroscience deposited at the National Library of Science in Washington. It's now up to 150,000. And it's continuing to rise. No sign yet of the asymptote. So we have lots and lots of data. We have no underlying theory. You heard about that this morning. You've heard about the absence of theory of how organic matter organizes itself into viable organs. So, lots of methods, vast numbers of methods, at all levels of organization, from subcellular, from molecular in some cases, right down to whole brain scale with pathology, EEG, imaging, behavioral studies, pharmacological studies, genetic studies. You know, many of these diagrams here have won Nobel Prizes because people have developed fantastic instruments, doing fantastic. I mean, everyone's going mad at the moment about these color-coded, what's it called? It's color genetics, chromogenetic genetic. What's it called? Brainbow. That's it, lovely. So lots of these things, but they're not connected. Because there's no scaffold, there's no theory, there's no blueprint, if you like. So the question is why? And there are two main reasons. The first is that people have got into a certain way of doing science. And in neuroscience, they're particularly ingrained in that. So they've not really used modern informatics, in order to help them to curate their data, to categorize their data, to classify their data, to cross-correlate their data. Everyone works in their little silo. So, this little list, which you know much better than me, these are 20 years of technical development. It's not very long. Some of these are industries that started from zero and now worth 100 billion. So, in 20 years, pushed by individuals and being used to affect our lives. iPhones, someone was talking, portable phones, someone was talking about, meteorology, someone was talking about. Some of you may have seen that nice article in Nature about a year and a half ago, which started off with all the information from the Hubble telescope from about 30,000 years post-Big Bang, and predicted using data mining on all the massive data that the Hubble telescope has produced, what the universe should look like now, and then compared to it, what it does look like now. And it was a remarkable correspondence between the two. The fact that we can predict the weather tomorrow, the day after tomorrow, and so on. Some of us, he's certainly lived long enough to know that if the weatherman said tomorrow will be bright and sunny, it was hailing and snowing and it was minus 10 degrees. It was as bad as that, wasn't it? They couldn't model the Humboldt current, but they now model the Humboldt current and many other things. So when this call for the flagships of enterprise and technology came out, there were a group of us who felt we need to move forward, we need to make a quantum jump here, we need to break the tradition, we need to move the culture on, and we need to bring informatics and neuroscience together. Now, the interesting thing is this happened at the EPFL, and the reason for that was that the EPFL was a little polytechnic, really, and about 15 years ago, a new director came along and said, we just have to focus. And he said, well, we'll focus on engineering, all from nano to classical, and life sciences. So there had already been the bringing together of life sciences in general and computing and engineering and so on. So it was a sort of natural development. And the second thing that I think that we need to think about is the philosophy underlying what's happening. And the philosophy that underlies the Human Brain Project and why it's so contested. So if you talk to the average Frenchman, any average Frenchman here? We've got no Frenchman. Oh, there's an average Frenchman, right. Very good, hello. So Descartes is very important in your life, isn't he? The Cartesian model of science is, and in fact, all of us subscribe to the Cartesian model of science pretty much. And this sort of in shorthand means I find a problem and I make a mental model of it. A mental theory. I think that A is Y, and that's why I've got Z. And then I mathematically express that. As someone was saying, that's the universal language of theories, has to be mathematics. And then I confront that theory with data that I collect. And I look at it, and I think, what's the error variance like? And then I think, well, it's pretty close. Well, let's just fiddle it a bit. So that's called parameterization. And then I'll fiddle it with a little bit more, and that's called optimization. And then I've got a fact. I've got a model which represents a function. The problem is that in the brain is not a function. The brain is controlling the whole of the rest of the body and interacting with the environment and everyone else there. There are multiple functions. So if you're going to model a single function in isolation, you know a priori philosophically that you're stuffed. It's not going to mean anything because that module will be interacting with all sorts of other modules throughout life and in different ways at different phases of your life and in different ways in conditions of health and in conditions of bad health. And indeed, you have no way of predicting it because you don't know about all the rest. So that's the big problem. That's why neuroscience at the moment is a bag of facts rather than a body of knowledge. Now, the radical change of methodology that was suggested by the application to the European Commission was that we should begin to use a different sort of modelling, a modelling that was led by the data. And the reason for that was that there are now vast amounts of data in research database, all sorts of places. And they're of many types and from many modalities. And again, we heard this morning how interesting it is to bring different modalities and many types of data together, because you can begin to enter into the world of complexity and of non-linearity. You know, when I was trained as a doctor, and still with people who do standard clinical research drug trials, we used and we use linear statistics that depends upon P. And if you're really lucky, they might know about T, and anyone knows about Z gets top marks in the class. And that's it. It's the sort of thing that kids at the age of 12 are learning now in school. And that's being used currently. So there was a whole flood of new mathematics came with the computing power, the whole flood of new statistics coming out. There was machine learning, which has had massive impact over the last 5 to 10 years. There's all the big stuff now about deep learning, conscious learning, and non-linear relationships in a large body of data can be exhaustively looked at, and the relevant, important, interesting bits can be taken out and used as hypotheses for further classical research. So essentially, it's saying if you're dealing with an organ which has an unknown number, but probably close to infinity, of degrees of freedom, with a mind that only has 4 degrees of freedom, X, Y, Z, and time, you're as likely to find out something about how the brain works as a group of monkeys with typewriters are likely to produce the works of Shakespeare. So what you need is help. This is the classical philosophical paradigm. How can the brain understand the brain? It can't. It needs a calculating machine as well. And other brains. So it's multiple brains, multiple calculation. Calculating machines understanding a brain, an average brain, the theory of the brain, the framework, if you like. So it's a machine, it's a methodology that tries to extract the hypotheses from real data rather than extracting the hypotheses from the imagination. That's essentially the difference. And that's why it was very important that the simulation model work. And in essence, this is what it was. The idea is that you can, as again was shown this morning, you can show various functions at various levels. So this is a synaptic level, this is a very large complex molecule, which is an ion channel. This is the level of individual neurons. You could describe the behavior of their interactions and their morphologies and so on with equations. One of the most commonly known, because it's even taught in 6 forms, is the Hodgkin-Huxley equation, which shows how conduction down a myelinated nerve cell goes in a saltatory fashion. That makes it go so fast, 60 meters a second. These equations can be put together with statistics. And there's a vast amount of knowledge from classical anatomists showing you that in the 6 layers of the cortex, there are various types of nerve cells, there are various distributions of connectivity between the nerve cells. You can measure these. These have been measured, these have been reproduced. So all these constraints can be brought in together with the mathematics that explains each of the individual functions, and you can begin to build models such that, be it anatomically or physiologically, your in silico model, which is not designed for any one function, but which is totally generic and based on data, behaves in the same way as a real piece of tissue, either in the way it's structured, looked at under the microscope, described in mathematical terms, or in the way it functions. You put in a burst of electricity, and at various rates, for example, you get various responses, be it in silico or in the living tissue. So this is what was the source of enormous contention, that models are data-driven, they're expressed mathematically, and they're biologically validated. And the most common criticism was simply, it is not possible. Well, the people who did the Genome Project had exactly the same sort of criticisms 15, 20 years ago. And this shows that it is possible. This is a paper that came out in Cell. It came out on the 6th of October, 2015. And essentially, it models the microcircuit, the mini column, if you like, of a rodent brain. And it generates a large number of hypotheses. Now, I'm going to tell you a little apocryphal story about this paper. The paper is 36 pages long, and there are 20 pages of online additional material. And there are 15,000 individual neurons with their anatomy and their physiology and their ion channel physiology published online, which you can use any time of the day or night you like. So this is a massive resource as well. When the paper was sent off to Cell, there was a lot of hoo-haing, and I've never done it, it's too big. And so Cell, eventually the editor said, we will send it to the 4 top people in the field, and we'll see what they say, and we'll take it on that basis. So the 4 reviews came back, and they were all positive, but wanted more experiments, wanted this, wanted that, wanted the other. So one of the guys in the lab took the 4 reviews and ran it through some textual data mining and discovered the identities of the 4 reviewers. Then they went to the literature, and these are all top guys, and they found what were their major contributions in neuronal physiology. Some of the things had taken 10 years to do, or experiments had been done for 4 or 5 years on tiny pieces of brain, nowhere near this size and so on. And they went to the model, they introduced the input parameters, they got the outputs, and they were the same. So within half a day, all the results had been reproduced. So this clearly shows proof of principle. Now, whatever else it shows proof of principle. And if you've been listening carefully to the talk previous to mine, you will understand that doesn't mean this is the model. It means that in a process of iteration, as one goes on and on and on and on, this model will get richer and richer and richer and richer, and eventually it will represent reality. Much more faithfully than it does at the moment, but it does a damn good job already at that level, starting with ionic channels and the cellular distributions and their connectivity matrices, going up to the whole full integrated column of about 300,000 neurons. This paper had 86 authors. This is like a physics paper. Unheard of in neurobiology. Unheard of in clinical medicine. So this is where it's going. It's a big culture change. It's a really big culture change, and it's going to take a lot of time. But it's going to depend on you guys, because we're too old. We can't change. Whereas you guys know the basis of these things much better than we do, and you're much more likely to succeed. Right, so that's a bit of background. Now, I'm an imaging guy, all right? So for me, informatics federates data. Now, I'm an imaging guy, but I'm also an absent-minded sort of guy. I left my computer in the security part of Charles de Gaulle last night. And so I've been given this computer, and I've got my stuff off the web. But unfortunately, there are 2 videos here, the top 2, which are not moving, but that doesn't matter. Essentially, they federate data because they encapsulate it in these icons. These icons are as numerical objects as a matrix of numbers. They just, you can capture their significance more easily through the visual system. So usually you see the brain turning around, that's an MRI. You could strip off the skull, you could look on the inside, the outside, up, down, you can take out little bits and everything. The one on the right-hand side at the top is one invented in Austria. I brought it because it's an Austrian invention. This is from Technical University in Vienna, who were the first, not the Americans, the Austrians were the first to put the rat brain in special oil for about 3 weeks and then to shine a monochromatic light through it, and to see, without cutting the thing up, all the cells. So you can see all the cells here in the where are you, little arrow? Little arrow, come back. Damn you, little arrow. She's disappeared. It's like a woman. There it is. So there you are, you see. Lots of cells, lots of pyramidal cells, and then you can see the axons going down into the hippocampal layers. This is the white matter. This is more neurons. So again, this allows you to picture how the connectivity works, and it actually allows you to make measurements. And this one is a really, really fascinating one because it's a hypothetical image. It's an image that starts off very small. That's the rat brain. And then it becomes the human brain. So this is an image that's trying to get across the temporal evolution of the brain over the last 2 million years. See how the different parts grow in relation to each other. Of course, it can be criticized because a rat is a current species. We don't know what the rat of 2 million years ago was like. But it gives you an idea of the sorts of ways you can manipulate images in order to federate the data. To federate data and to bring data together and make it comprehensive. It also integrates data. So in my field, one of the interesting things we do is to follow connections in the brain. And we do this using MRI, so it's totally non-invasive, and it uses a property of diffusivity of water in different compartments in the brain. So it's called tractography, follows the tracts. And my last PhD student at UCL, when I was there, a very nice guy called Christian Lambert, who's doing great things since, wanted to discover the connections of the subthalamic nucleus. Now, this is a nucleus deep in the brain where surgeons put electrodes in Parkinson's patients who are trembling like this. And then they switch on one side, and they go like that, and they switch on the other side, and the patient's like that. So it is absolutely dramatic. I mean, you have to go to the theatre to see this. It just happens like that. But it's a tiddly little thing. It's sort of just about the volume of a milliliter. And it has different parts. It has an anterior part and an external part. And he wanted to start understanding how this electrical activity in this tiny region affects something as major as a tremor and movement and so on. So he wanted to work out all the connections in the brain. So he said, well, where shall I start? I said, well, we've just been hearing about these guys at EPFL who have been using data mining. Why don't you data mine all the papers between, say, 1947, when the National Library of Science was started, and 2011, which was about the time he was he was beginning the experiments, which mentioned the subthalamic nucleus and some other named area of the brain, wherever, in humans. So that's what he did. And this is the diagram, this synthetic diagram after essentially 6 hours' work of doing that. And it shows in thickness the number of connections, in red whether they were afferent or in blue efferent, in light blue mixed, from the different parts of the brain, many of which are cortical, but some of which are also subcortical. So you can see the connectivity is really very large. Now, we've heard about reproducibility, so at least you could say the thicker ones of these, perhaps not some of these very thin ones, but the thicker ones of these are credible. Then he went to the MRI machine and he generated these data. So these are the parts of the brain looked at from the side, looked at from above, and looked at from the front. Okay, so here are the amygdala, for example. And these are different parts of the brain, thalamus, caudate nucleus, putamen, etc., etc., etc. And these are the connections. Now, the color code here shows that in blue, you have the connections with the posterior aspect of the subthalamic nucleus, which is mainly interested in motor things. So the sorts of things that the surgeon is treating. And you can see that they are quite distinct from the green and red ones. And they go through different parts of the brain to the green and red ones. So this is a system that you're beginning to pick up in vivo, in a normal human, using data that you've collected from anatomy that many, many scientists have put together as text into the National Library of Science. In red, you have all those that have something to do with emotion and memory. And you can see they have very different areas. And then there's the green ones, which project to those which have to do with the emotive parts of action as well. So this is a way in which you can start building on what other people have done. And this is a very important thing. You know, most of the time we're trying to compete with other people. You mustn't see my data because you might get something out about it before I do. Whereas what this is saying is start sharing your data, because you'll find out a lot more. Not only will you find out a lot more, you will integrate a lot more, so your power will go up, your sensitivity will go up, and you'll get more reproducible data. And the more data you put in once you've got your model, the more you'll see what the fluctuation in the error variance is. And when you get a nice, stable error variance, you know you're home and dry. The other thing is that informatics helps us classify data. This is an experiment I did now some time ago, in 2007-8, where essentially we were tackling this issue of how do we diagnose Alzheimer's disease more accurately. Now, what I haven't told you is the fact that those people who have done big studies of this, using the end-stage pathology of Alzheimer's disease as a cause for dementia, are uniform in saying that we're wrong about 30% of the time. Some people say 40%, some say 25%, but it's a mean of about 30% of the time. So what this means is not that people can't diagnose dementia, because that's relatively easy to diagnose, but they can't diagnose the cause of dementia. And there are many causes. Everyone knows about Muhammad Ali, that was trauma, right, for his Parkinson's, very early. There are infections that could cause Parkinson's disease, post-Spanish flu, encephalitis, which caused a sort of Parkinson-type syndrome. You remember that film with the Oliver Sacks thing? Then you could have degenerative standard Alzheimer's disease. You can have one of these behavioral variant frontotemporal dementias. You could have another type of frontotemporal dementia. All sorts of diseases. Those are the ones we have to diagnose because each has a different mechanism. So each really does need a different drug, if it's drugs that are going to help. And each one of them eventually we should be able to diagnose preclinically because of the redundancy. So there's a big task there. So we've been playing around a bit with machine learning and with support vector machines. And I have quite a big network. So I wrote to lots of my mates and I said, do you have the scans of people who have subsequently died, who you thought had Alzheimer's disease, and who turned out to have Alzheimer's disease and nothing else post-mortem? So I got about 30, 40 of those. And I also asked them, do you have the scans of people who have subsequently died, where the scans are reasonably close to when they subsequently die, where their brains have been absolutely normal, no evidence of subclinical Alzheimer's disease? Right? Because at the moment, when you create a study to test a drug for Alzheimer's disease, you construct a cohort of Alzheimer's disease, you've got 30% error variance. You then construct a cohort of normal people, age and sex matched, and et cetera, et cetera. But there's an error variance there as well, because there's lots of compensated Alzheimer's there, quite likely. Okay? So you need massive cohorts. That's why it costs so much. So now I had real Alzheimer's and real normals of elderly. And we constructed one of these support vector machines. And we first took some Alzheimer's disease patients and controls and their clinical results, and we found that there was quite a good, actually, correct classification of 20% error there, terrible sensitivity, reasonable specificity. But this took, a week's worth of hospitalization, a neuropsychologist, a radiologist, all sorts of people. These, one scan, support vector machine, trained it, and then tested them. 95% correct, repeated, 93%. Massive sensitivity, and really very good specificity. And the interesting thing then was to go on to refine analyses using these this support vector machine, because the support vector machine now constitutes essentially a model. So now we went to a database called the ADNI database. You may have heard of it, you may not have, but it's a massive database which started out at UCLA. I think it's in the University of Southern California now, which has spawned about 400 papers over the last 10 years. The group has been collecting scans, collecting clinical data, collecting genetic data, making it freely available. One of the very first open, freely available databases. They're in their second phase now. The first phase were about 600 patients altogether. So we went to the ADNI database, and these are the ADNI diagnoses. So we found 20 who had Alzheimer's disease in the ADNI database, and 20 normal controls, healthy controls, in the ADNI database. And we then tested them against our model, the support vector machine. And we found that 5 of the 20 were misclassified. Now, you have to decide who's doing the misclassification. We believe it's misclassified clinically because we have the model which shows us that we have the sensitivity and the specificity and the accuracy of the SVM, which is close to 100%, whereas the other one's down at 70-80%. So, and then the more interesting was that some of the healthy controls were also Alzheimer's on the basis of their scans. So these presumably are those compensating Alzheimer's. They have a pattern of atrophy of the brain which resembles that of Alzheimer's disease, but they don't have the disease yet. We just started getting data out to show that they transform into full clinical Alzheimer's disease much more quickly than healthy controls that don't have that pattern, this group here. So again, informatics can refine analyses. So when we were thinking about future medicine and the medical informatics platform, essentially our thoughts were in terms of syndromic diagnosis, which had reached its limits. We wanted now to use all the new information that had come in over the last 20 years from the Genome Project, from all the modern neuroscience, even though it was fragmented and atheoretic, from modern clinical neuroscience with all this imaging stuff that had been coming in since 1972, from the information technology that I showed you a list of, and from modern mathematics that all spawned, to give us groups of patients who were identical in particular vectors of clinical, physiological, anatomical, hematological, chemical, psychological data. And those people who had the same abnormalities, we would call having a disease with a particular disease signature. And we believe that that would be a much more mechanistic diagnosis than the syndromic diagnosis I've been talking to you about. Now, the other interesting informatics side of all this is that there is a hidden wealth of data which is totally unused, available to all of us to try and do this. And that hidden wealth of data, locked up, is called hospital databases. It's hidden and locked up because of privacy. And that's why people don't use it. And the law requests that hospitals keep these data for 15 years. That's a lot of computing, a lot of memory, a lot of storage space, a lot of salaries for nothing. Actually, it's not quite for nothing, because there are a lot of lawyers who like to see these things when their clients come and ask whether they can take a given doctor to court for malpractice or something. That's just about all it's used for. Oh, you might say, but we follow patients up. Well, we do, but only about 5 to 8% of the data are ever used again for that. So it's a really wasteful thing, but imagine all those data, and we just don't use them. So that was a really big stimulus to us, because if we could do that, and if we could do it over a lot of hospitals, we could catalyze a major collaboration between hospitals for federating and integrating knowledge and data, and start creating IT-based encyclopedias or Wikipedias or whatever you like to call them, where each part of the brain would be tagged with physiological knowledge, disease knowledge, connectivity knowledge, knowledge about the genes that control its development, and so on and so on and so on, allowing to analyze and derive really biologically grounded brain disease signatures for this new diagnostic classification. So, what are the data sources we have available and what are the challenges? Well, there are two big ones. Well, actually, there's one small one and one very big one. The clinical data in every hospital in Europe, at any rate, contains clinical data. It's incomplete. It's not the same for every patient. It's really messy. It's not structured. It's not standardized. It's not clean. Very protected for privacy and very protected against corruption. Very important. You can't corrupt the data because otherwise if the patient came back and you compared his current data with his previous data, if it had been corrupted, you'd be up the creek. The other set of data we have are research data. Now, these are much more structured, particularly if they come from pharmaceutical companies. But pharmaceutical companies hide their data for commercial reasons. But then it just needs a little bit of thought. So I went up to this pharmaceutical director and I said, I want some of your data. He said, oh, yes. So I said, but I want it from your failed clinical trials. He said, oh, well, that's quite a lot of it then. And I said, I don't want it from all the subjects. I want it just from those who were taking the placebo, so that it's not contaminated by drug effects. Oh, well, there's no commercial interest in that. So we immediately got 400 sets of data. Now, those are really well structured. You could do fantastic stuff with that. So what we conceived of, the research data, the other research databases, our research databases. We were so precious up to 10 years, 15 years ago. But we had to really not allow anyone to see them. You could hardly let them look at the computers, could you? Because they might steal our ideas by looking at the data. Well, of course, that is changing very fast since the Allen Brain Atlas thing, since the NIH demands that anything that's funded by NIH be open access. Wellcome Trust is now doing that. Since people realize that it's us that pay for those data. I mean, if I give you a grant as a head of a funding agency, you do some work, the data you've got from the people who have helped you, data that belong to the people, and belong to the taxpayer who has given me a large sum of money to give out as grants. So it's all taxpayers, it's all population, it's our data. They can't be, and anyway, the data never win the Nobel Prize. It's the idea that wins the Nobel Prize. The data are used to support it. So we talked to the Allen people. They now virtually take the data off their experimental machines and put them directly onto the web. It's not quite like that, but it's virtually like that. And so they publish 3-4 years later on data they put on the web. And they've never been gazumped for anything. And on the other hand, the ADNI people, they had masses of citations because they're on every paper. So there's a little interesting game played there, but getting credit. Okay, so the ethical challenges, that was the big thing that everyone said would make this fail. So at the moment, what hospital IT directors do is they depersonalize, and they call that anonymization, which it isn't, of course. Famous paper in New England Journal again, which said that if you had, even in 1 million subjects, the date of birth, the genome, and the address, you could, no, not the address, and the postcode, you could identify individuals. So that's clearly not anonymized, even if you had no names, no other things available at all. So we wanted to get on with this quickly. So we just simply said to ourselves, well, what really is of interest to us? What's of interest to us in the first place is to get these disease signatures. Disease signatures talk about groups. Group data is usually aggregated data. So we said to ourselves, what we will do is we will work only with aggregated data. Now, if you aggregate the data, currently, the cryptographers tell us, if you aggregate data from at least 10 subjects, it is anonymized. If it's anonymized, it doesn't fall under privacy law. It's excluded from privacy law. It also doesn't allow anyone to come back to you if you're one of those 10, or one of those 1,000, or 100,000. So that was the first solution. And so people said to us, well, what if you discover something? Well, they said, well, if we discover something that might make a difference to people's health, first we'll tell them about it. And secondly, we'll give it to the doctors. And then the doctors could discuss with the patient, under the law that regulates doctor-patient relationship, which already exists, whether that vector of data can be compared to the disease space with its disease signatures that defines the new categorization to see where it lies closest, and then use it to diagnose or to manage or to prognose or whatever. So that was also accepted. And the third thing that was very important was that we have a lot of ethical committees locally which have lay people on them in the whole of Europe. Another advantage of Europe is, of course, we've got a lot of socialized medicine, so that we really are the taxpayer, we really are paying for all this medicine. My data is as much yours as it is mine, only an infinitesimally small part for you, all mine, except for the infinitesimally small part that you each also own because you've paid for it. So these local ethics committees have given a lot of value, a lot of credibility to medical science. Indeed, all the sociological analyses and so on always put doctors top of the trust list. And I think that's a jolly good job too. So these ethical challenges have been debated now for 5 years. There's a law going through the European Parliament about data privacy, which has been substantially changed because of the discussions that we've had, and will allow medical science to continue, and will allow this sort of work to continue, with some discussion still about what sorts of consent can be obtained, but that's a trivial thing. So now with all that information together, we sat down in SP8, medical scientists, basic scientists, and informaticians, and we started designing a system which would allow us to go out into the databases of hospitals around the world and collect data for medical research and for medical care. And the analogy we used was Google. I type in a question, my question goes out, finds what I want, and sends it back to me. Now, it's not a good analogy, but this is it. So the first thing to remember is that this is the hospital security wall. It's the firewall. This is the primary database, untouched, unmoved, uncopied, well, copied but uncorrupted. The informatics officer always makes backups. Okay, so we take one backup, and that's what we work on. So make the files uniform, which is standard now. We do this in real time, and we depersonalize using standard techniques, currently top-of-the-range industrial standards that are in place already. We then pre-process the data. Depending on what it is, we denoise it by smoothing, we standardize it to 0, +1, -1, we anatomically normalize it into standard brain space, which we've been doing in imaging for 20 years, or in sub-millimetric resolution, all automated, numerically normalized, and we start creating links between the different words, different languages, and different hospitals and different schools call the different diseases and the different organs. And it's the sort of thing that Google has spent the last 5 years doing for Siri, so that you can work out and identify the various words that people use and the various pronunciations and in the various languages. And that is then our database, the HBP database, constantly updated, from all the data from all people with any neurological disease. That includes all of psychiatry, neurosurgery, and everything. And then it's there. In the context of a federating infrastructure and talking to and receiving queries from a unified portal. This is the HBP unified portal. So the portal is where the scientist sits. He sends out this query. It goes to as many of these hospitals as it identifies data existing for. On the raw data, it selects those data, it aggregates them, it encrypts them, it filters them on more for personal data, and sends them back to the portal. And then when it gets to the portal, it aggregates again all the aggregated data from all the sites. And people said to us, well, the statistics of this is going to be really messy. Actually, the statistics was the easiest thing, because you just apply Bayesian principles here, because you're getting the first set gives you Bayesian, and then the next set, and you just add it all up, you get perfect statistics out of that whole thing. Throughout, of course, we store the provenance at every point, who used it, how it was used, how it was processed, and so on. So that in fact, there may not even be a need to hang on to the result, because you could always call it back. On the more accumulated data, possibly making it more and more refined, able to look at what the variability is over different parts of the year and so on and so on. And the results are then published and whatever. We've now got 5 hospitals. We've got the infrastructure is in the CHUV in Lausanne, is in the Neguada Hospital in Milan, is in the University Hospital Neurology Clinic in Freiburg and Brisau, and the French Salpêtrière and Oxford and the Institute of Psychiatry in London are on the list, and we're just in the process of sort of sorting out. And we have a list of reserves if any of those sort of drop out. And in the current situation, people are using the infrastructure and the analytical tools locally, getting used to them. And the idea is that in the end, everyone will have the unified infrastructure, but will be able to design their own apps, if you like, for analyses of a particular type, depending on their hypothesis and so on. So the queries are distributed. All the data is seen as just one set of data. There's no localization information. You could choose what data you want to see. You collect the results. You do the queries on raw files as native storage, so you have no database, no loading into databases. You have retained data formats, files and scripts, so you could use historical data. You don't need to change anything that has been used in the past, because it's the raw data that count. Anastasia's group have been doing performance and optimizing speed of data access and other such benchmarks, and you can see that the no database of the solution that they're using, which is a virtualization solution, if you like, outperforms a lot of standard databasing, doesn't need the standard database formats and prescribed ways of loading the data into the database in order for it to be addressable in a standard sort of manner. And this allows us to explore the data for the disease signatures, to data mine in real time in a continuous and iterative manner. We haven't started that yet. We've done that as little parcels. I'll show you one of them. Visualizing the data for epidemiological and health services research. I'll show you a little bit of that. And then hypothesis testing and clinical trials. Trying to go across the scales from behavior back down to genetics. The power of, I'll leave that out because I'm getting on. So, data integration. We got these 5,000, roughly 5,000 data sets from the French, who were remarkably good. This is a research database run from Toulouse, from Bordeaux, and from Nantes. Lots of brain imaging, lots of clinical scales and measurements, lots of genotyping with anything from 500,000 to 1 million SNPs in some of the patients. Proteins in the blood, amyloid in the cerebrospinal fluid. So, again, data coming from different labs, different formats, not complete, repeated in some cases, and so on. Integrated, actually in an Excel file, but we don't do that anymore. Color-coded, and here is the first set of results in an aged population. And what you see are these coloured discs, and the red coloured discs are discs that identified people with cognitive decline. And the blue coloured discs are people who apparently are normal. And it's very clear that there are more than one set of normals, which now you would be expecting, because you'd be expecting, firstly, that the normal pattern would change with age. Secondly, you would be expecting that there would be compensated Alzheimer's or other diseases present, people who are normal but who already have the pathological mechanisms in place. The big central one there looks like Alzheimer's disease from the point of view of it's associated with genes, the ApoA eta4, which increases your chance of getting Alzheimer's disease if you have the homozygote of the eta4 variety by 24%, which is the highest risk factor of any association for Alzheimer's disease. It has a pattern of atrophy, which is the specific one that we got out of the support vector machine for Alzheimer's disease, and a number of other features. The little black dots are genetic information. The little green dots are patterns of atrophy. You could see that there's separation of different genetic types around the different clusters. And these, we believe, are at least very indicative of the possibility of being able to identify subgroups which would constitute really biologically based diagnoses of very homogeneous groups of people. Now, the caveat here is there's only 5,000 people. So to do proper data mining, you need 1 million, 2 million. You need to really get that signal out and bash the error down. But it's very significant. And you can visualize it in all sorts of ways. So this is one way that's been developed, which is nice that it shows that there are clusters of these, rather like a little solar system, usually based around a gene, but sometimes around a type of atrophy. You can see there's some very unique, like orphan diseases almost, but there's a little group of people in there that has this pattern of genes and this pattern of atrophy. It's called a rule, a vector, which they all share, and it's just another way of visualizing. And then we're playing around with medical epidemiology. So here are some diagnostic categories from the standard symptoms and signs diagnostic catalogue. You can see them here. That's the general disease. Those are subcompartments of the disease. These are the number of patients that came into the SHUV with each of the diseases out of the 57,000 patients with brain disease that we saw. You could start looking, most of this is actually done in real time. You can look at mental and behavioral disorders, look at which of them are Alzheimer's, which of them are another disease. You could look at all the names that are given to dementias of various types. You could associate that with sex, males and females. All sorts of visualizations that I won't bore you with any longer because I'm well over my hour. So there are plenty of things that can be done, and we think we now have a platform that's almost ready to be distributed internally. The 5 hospitals will be our internal R&D developers. We have a number of work packages around the platform, which include those that relate to data capture, data query and data mining. I talked to you about this, said this probably is going to be the hardest work and may need vast numbers of people, probably in countries where we don't pay them very much doing the ontology, but we're thinking of ways of doing this, thinking of ways of doing this informatically as well. There's an awful lot done on ontologies already on the web. Workflows, curation, managing the federated data, which we're leaving with the hospitals. So it's distributed in that sense. It doesn't need any more people. Data acquisition and federation, it eliminates the need potentially for data warehousing, unless you might need 1 or 2 data warehouses for specific experimental results which don't come from the hospitals but come from other places and where you want to store them together. But we can't think of very many examples of that. Then we've got the users and the community outreach. We're even thinking of involving patients in curating the data. Patients love being involved in doing things with their own data. And then the whole scientific coordination, and then the medical intelligence tools. We've got some big competitors now. The AI group at Google is going full tilt into medical informatics. Apple are playing around with it, all sorts of other people. But we think we got a bit of a head start. We've got a unique strategy, and it fits into the initial idea of the 6 open access platforms, of which now you know why medical informatics is one. The other thing that medical informatics does is it will produce, of course, configuration files where for each of the variables that we potentially can look at, we will have abnormal values depending on which disease signature there is. And then the high-performance computing, neuroinformatics, and the simulation platforms will be creating the theory for the normal brain with configuration files, with lots of variables, with the normal values in. So the potential for reverse engineering the normal with the abnormal and seeing whether one produces changes which propagate throughout the whole system that are identical in real life and in silico is there. So it may be one of the better ways of thinking about eventual and final validation. So we continue to think in terms of future neuroscience, future medicine, future computing, though that's proving a little bit difficult at the moment. And we're moving into the next phase, which is called SGA1, with the 5 academic partners that are receiving the Federation infrastructure now, and where we are essentially, having deployed all this just at the CHUV in Lausanne, we're now scaling up. And if we can scale up, and scale up in time as well as in volume, which is what Anastasia's group really feels they can do, we will, at the end of that phase, tender for commercialization, open access to researchers, and try and begin to conquer the informatics world of brain medicine in order to make a leap forward and try and start dealing with the dementias and the schizophrenias and all the other things. This is a very collaborative, cooperative, non-competitive, focused, mission-centered, single vision project in its conception, and lots of people need to defend that idea. That it doesn't dissipate into just, lots of people continuing to do what they've always done. And your voice can be important in that as well. And of course, you can get involved. So I'm open to questions. I've gone over time by 11 minutes and 3 seconds, and I'm very apologetic.